The genes for type 1 diabetes have been found. Maybe not the genes themselves, but at least the loci where they reside. This work has been completed by far and large. You see that more recently we are not hearing about new discoveries. But most of those genes have functions that are still unknown. And the first one discovered, HLA, still has a lot of secrets. Generally, HLA molecules, both class 1 and class 2, present antigens to T cells. Class 1 to CD8 cells, class 2 to CD4 cells. Those could be antigens that come from outside, like you can imagine in celiac disease, the antigens come from the outside and are processed, the peptides are modified, and then presented and may cause autoimmune diseases. Those could be also autoantigens that are released from our own cells under some special circumstances like infection or stress. Those 5 HLA molecules, DR, DQ, DP, A, B, and C, have been found to be associated with type 1 diabetes. But it's now possible to dissect which amino acids in the functional piece of the molecule called groove, which presents the antigens, which of those molecules, which of those amino acids are critical to susceptibility or resistance to type 1 diabetes. You may see some yellow dots in different places of those molecules. Those dots present the critical amino acids that confer susceptibility. Interestingly, the variants that confer resistance are located in the same places, just different amino acids. The blue line shows the resistance DQA1 epitope, which is the only resistance epitope different on this slide. So this molecular structure of HLA is only now being discovered with better tools and allows us to go beyond alleles, haplotypes, and genotypes and look at the function of HLA, what it really does in terms of antigen presentation. Here you see that a person who is a homozygote for alanine in position 71 of DRB1 is highly protected from diabetes, but having 1 or even worse 2 lysins in this position code for a number of susceptibility alleles, including the classical DR3 and DR4. So it's not a single allele or haplotype that determines HLA susceptibility or resistance. In this paper, the authors developed a score. They assigned 2 points to each of the 6 potential proteins in HLA involved. And you see that age of diagnosis of type 1 diabetes is inversely related to the number of susceptibility epitopes present. So this is how this HLA molecule looks like. This is DQ8. And here it's caught in the act of presenting a piece of insulin, a classical autoimmune peptide including amino acids 9 to 23 from the chain B. Now, in many experiments, this phenomenon has led to autoimmune destruction of islet cells in animal models. we can now prevent DQ8 specifically, from presenting insulin or anything else in this groove if it's in contact with certain small molecules. For example, methyl dopa, which is a drug for hypertension, Aldomet, has this property of blocking presentation of insulin peptides in the groove of HLA-DQ. Now, the story gets more complicated because it's probably not the native insulin that is presented in the context of type 1 diabetes. It's something modified, post-translationally modified peptide. And researchers are playing now in the laboratories trying to modify insulin peptides to see which ones have properties inducing autoimmunity and which ones may be helpful as potential vaccines. And here's an example of the same 9-to-23B peptide with one substitution at the position 22, arginine for glutamine. In experimental studies, including newly diagnosed type 1 patients, shows very interesting properties of inducing IL-10 response. And as you see in this insert, inducing also a nice subpopulation of Treg cells. So by playing with those peptides, we may discover not only what causes type 1 diabetes, but also how to potentially prevent it. But we still don't know what causes type 1 diabetes, and there's a very dynamic new field of post-translationally modified peptide studies. Here are several examples of T-cell clones that have been isolated from NOD mice. And for a long time, we didn't know what those clones are actually reacting to. It turns out that they are not reacting to insulin or chromogranin or other islet-specific proteins. They are most reactive to peptides that are hybrids, contain a piece of C-peptide sequence on the left in the red, and a piece of chromogranin in the first row. So how does it happen that those hybrid peptides are created? It turns out that they are created all the time, not only in the islets, but in many places where proteolytic action is robust. So, to summarize HLA, it explains more than 50% of type 1 familial clustering. The risk of type 1 and the age of diagnosis depends on a combination of a number of HLA epitopes. Small molecules can be explored in terms of blocking the action of, or potentially enhancing the action of HLA. And the next frontier here is hybrid peptides and mimotopes, modified peptides that can have therapeutic or preventive properties. What about non-HLA loci? So the most interesting development in the past couple of years was the immunochip consortium, 200,000 SNPs on one chip, SNPs for loci associated with not only type 1 diabetes, but this long list of autoimmune diseases. And for type 1, 25,000 patient samples have been already analyzed and a large number of controls. So I'll show you the results later, but some of the monogenic forms of type 1 diabetes, like diabetes in the IPEX syndrome, where 80% of children develop insulin-dependent autoimmune type 1 diabetes, some of those monogenic forms have been already cracked. We know the gene, we know the variants that cause the disease, we know the function, We even know the clinical course fairly predictably, as listed here in the bold on the left. And we even know how to treat those diseases. This is actually my patient that I saw one night in the NICU. And it's a success story. This is the only type 1 patient that we have cured. DR3/4, HLA-DR3/4, autoantibodies, absolutely type 1A diabetes, but after the bone marrow transplant, this kid went into remission and is now in total insulin remission for 2 years. So I wish we had the answers for garden variety for the 99.9% of type 1 patients, how to cure their conditions. But it's possible. Now, those 40-some non-HLA loci that have been already mapped, some include up to 25 different genes. Remember, so we don't know for many of them what the gene is. We still have to find out which gene in those loci is associated with diabetes and what it does. So this is the next frontier in this line of research. To summarize, fine mapping analysis, such as those using immunochip, identified a total of 42 non-HLA type 1D risk loci. And there are some new ones identified, like the one in the interleukin-7 receptor. Interestingly, only 4 of those loci listed here, including PTPN22, there are only 4 of those loci where the most associated SNP is a non-synonymous coding variant. So many of those additional non-HLA loci are likely to be enhancer sequencers, not promoters of the genes, and most of them, we think, are involved in immune function in T cells, B cells, and stem cells. So we've covered genetics, but obviously genes do not explain why people get diabetes. There's this environmental part with which genes interact. How else would we see such a rapid increase in the incidence of type 1 diabetes, which is doubling every 20 years? There has something to be different in environment of our children today than 20 or 40 years ago. So viruses have been the prime suspect for a long time. Now the list is getting a bit shorter because we are getting more vaccines. So we are preventing natural infections, and rubella, rotavirus, or mumps should not be anymore a suspect for type 1 diabetes. There is also very little new evidence for some of the other viruses. The only virus left standing today is enterovirus. Would enteroviral infection be consistent with what we see in the pancreas? So this is what we see. The pancreas is not affected in people with pre-type 1 diabetes or with already clinical type 1 diabetes. It's not affected homogeneously. There are patches of disease. There are single lobules affected, like the one on the right in brown, where you don't have any functioning islets. You don't have any insulin cells. But next door to the left, you have a completely normal tissue with all of the islets preserved and no insulitis, no T cells. A little bit like vitiligo. So researchers from Norway were able to obtain pancreatic biopsy from 5 or 6 adults newly diagnosed with diabetes and found in a painstaking experiment series that about 2% of islets in those patients show signs, molecular of course, signs of enteroviral infection. 10% no, I'm sorry, 2%. What is 2%? Can it lead to diabetes? Well, perhaps, given enough time, 5, 10, 20 years, it may accumulate to the damage that we see at the diagnosis of diabetes. There is a very interesting hypothesis presented here as a cartoon, showing that if one gets infected with a normal enteroviral virus, the infection is cleared fairly quickly and does not cause to autoimmunity. However, if the virus is able to mutate and create a defective mutant missing some 8 or 10 terminal amino acids in the critical protein, then this virus mutant can detect immune surveillance to some extent and persists as a constant irritant, so to speak, to lead to autoimmune damage. Something to be tested in humans for sure. Observational studies are very confusing in terms of enteroviral infections as the cause of diabetes. Most of them, of course, are published because they show significant p-values. So it's interesting to think about those studies that have not been published and were negative. But nevertheless, I cannot make my mind up. Also, we've contributed to this field. If enterovirus triggers autoimmunity or promotes progression from multiple autoantibodies to clinical diabetes, or perhaps participates at both stages of the disease. So in summary, a presence of enterovirus in blood may predict development of type 1 diabetes in some cases. Evidence of persistent enteroviral infection has been demonstrated in islets of newly diagnosed patients, and we still need much more work to figure out if and how the virus can cause, specifically enterovirus, majority of cases of diabetes. On the positive side, routine immunizations or timing of those immunizations have been shown over and over again to not be related to type 1 diabetes. This is the message I would like you to take home to patients who may be asking those questions. Now, diet. Similar story, also a bit more consistent. What we know almost for sure with agreement from several prospective studies is that cow's milk is not, exposure early in life to cow's milk is not a risk factor. Exposure to cereal too early or too late may be a risk factor, and deficiency of omega-3 fatty acids in diet and in circulated blood may be a risk factor for development of islet autoimmunity. This is what we know from prospective studies. Now, there have been several very important clinical trials in this area. Unfortunately, all of them have shown no effect. The best, the largest, is still ongoing. It's trigger study. And you've probably seen the results published in JAMA last year, showing no effect whatsoever of substituting cow's milk formulas with nutrinogen or hydrolysate of that milk on the incidence, on the development of islet antibodies. The study keeps going to look at diabetes at the endpoint, so it's not yet over. TEDdy is another large prospective study, international, involving Sweden, Finland, Germany, and the United States. We've screened almost 500,000 kids to find about 8,000 of those with high-risk HLA genes, and to follow them from birth for a number of infections and changes in diet, pretty much drawing blood from those kids every 3 months. An incredible commitment on part of the families, and now some of those kids are already 10, 12 years old, on the part of those kids as well to help science. Just one snapshot from Teddy. You know the story of probiotics? It's really hot here in Europe, especially Northern Europe. You can see the use of probiotics in infants by year. It's now more than 50% of Finnish and German kids who get probiotics as part of their infant diet. And in Sweden, it's growing. In the US, it's not yet present. So Teddy had a great opportunity to look at the potential association with exposure to probiotics and type 1 diabetes. And here it is. It looks, it has to be confirmed, but it looks like if you give any probiotic to a child, mostly in Finland or Sweden, in the first 3 months of life, it's associated with a much lower, 30% lower risk of developing islet autoantibodies. Giving it after 3 months of age or even more after 1 year of age makes no difference. There's a lot that we don't know. What resides on stuff that we eat, in bottled water, and in tea that we drink. We think all of that is safe, but it's been processed, stored, processed again, and has a lot of not only pesticides and herbicides, but also molecules produced by bacteria and fungi during the process of storage and transportation. There are at least some anecdotal pieces of evidence that some of those mycotoxins may cause diabetes in animal models. And by the way, do you know what is the first vegetable Finnish kids eat? Potato. I hope not as scabious as that one I showed. Now, there are still people who believe in accelerator hypotheses that obesity and insulin resistance causes development of islet autoantibodies and type 1 diabetes. When you look at a number of prospective studies so far, there's no evidence for accelerator as a cause, or obesity as a cause of type 1 diabetes. So, little evidence for cow's milk, possible benefit from well-timed breastfeeding, early probiotics, and potentially omega-3 fatty acids. Very little support for accelerator hypotheses, and perhaps we need to focus now on environmental toxins. Bacteria, gut bacteria. To make it short, you are probably reading already some studies coming from excellent groups, but very small studies on maybe 12, 15, 20 children followed prospectively, claiming that this type of bacteria or higher diversity or lower diversity may be preceding type 1 diabetes. The problem is the same as we had with GWAS, genome-wide association studies. You will get a lot of false positive or false negative reports initially, unless the study is really large. So Teddy is trying to do just that in this nested case control study with 40-some kids who have developed persistent islet autoimmunity and 400 controls with typing of more than 13,000 stool samples and 6,000 plasma samples to find any RNA, DNA, any life, sign of life that is not human in those samples, and track it back to the bacteria or virus that it came from. So the early data from Teddy shows that the diversity, bacterial diversity, may be actually higher in cases who develop diabetes than in controls. It's in contrast to some previous smaller studies. And clearly, when you look at the map, heat map, there are some differences in each of the 4 populations included in TEDdy in terms of associations between specific subtypes of bacteria and development of islet autoantibodies. So it's early. I wouldn't bet on any of the early studies. Even TEDdy may be wrong the first time, so we need confirmation. And we also need to include in those analyses all of the metadata like breastfeeding, introduction of different foods, and of course, courses of antibiotics. We have, at the end, to put it all together, and it's already happening. By we, I mean studies like DAISY, TEDdy, DIP, Baby Diab, measuring a lot of sequencing genes, measuring gene expression, measuring proteins and metabolites that precede development of diabetes. And we are trying to interface it with the environmental data that we are collecting from the families participating in those studies. Hopefully, this will lead us to the answer, what is the cause of type 1 diabetes? I mean the environmental cause. If you ask me today, my opinion may change next week, but this would be my hot list, what is hot and what is not. So enteroviruses at the top, followed by decrease in childhood infections and hygiene hypotheses, maybe microbiome, and then everything else is getting a bit more iffy, or has been shown in blue not to be related to type 1 diabetes. Thank you very much.